Assalamu alaikum. I'm Muhammad Nuaishir Rahman. I will present our work ScribbleSeg, segmenting spatial transcriptomics data using Scribble annotated histology images. Spatial transcriptomics, or ST, is an emerging technology to profile gene expression at spatial resolution, and it can reveal new insights into molecular aspects of tissue architecture. One such aspect is transcriptional homogeneity in tissue regions. Since ST data capture gene expression at spatially resolved spots, clustering the spots based on their transcriptomic profiles can demarcate transcriptionally homogeneous tissue regions. The goal here is to segment the ST data, similar to how image segmentation algorithms demarcate objects in images. Notably, most image segmentation algorithms are supervised, that is, they require training examples with segments demarcated a priori. However, such training examples are scarce in the realm of ST, requiring ST segmentation algorithms to be unsupervised. Problem statement: we are given an ST data of around 3,500 spots, and we are given for each of the spots, 1, the gene expression levels of around 30,000 genes, and 2, the 2D coordinates of each of the spots. Optionally, there is another input, which is, like in this figure, for a subset of the set of spots, some pre-calculated labels assigned by a human expert as scribbles, or assigned by another clustering algorithm. Initially, each of the spots is unlabeled, and our goal is to label each of the spots into clusters given these 2 or 3 optional, 2 or optionally 3 inputs. Some research questions and objectives. Lahnemann et al. presented 11 grand challenges in single cell data science, and the 5th of these challenges is finding patterns in spatially resolved measurements. Our objective is to solve this problem. Some research questions are: how can we solve this problem with an unsupervised or supervised approach? Another question can be incorporating minimal input from human annotators. Can we perform better than state-of-the-art models? Here's some related works. Bayes, which is a state-of-the-art fully Bayesian statistical method that uses the information from spatial neighborhoods for resolution enhancement and spatial transcriptomics data and for clustering analysis. Bayes resolves tissue structure that is not detectable at the original resolution and identifies transcriptional heterogeneity inaccessible to histological analysis. Bayes incorporates MCMC methods like Gibbs sampling and metropolis testings to implement these. But Bayes does not have the capability to incorporate prior knowledge about the spot labels from expert annotator. In the case of human DLPFC data, Bayes uses output of a non-spatial clustering algorithm named mclust, and then smooths the data as shown in this figure. SMEB is another state-of-the-art model that does clustering with hidden Markov random field using empirical bias. SMEB is not only computationally efficient and scalable to large sample sizes, but it also is capable of choosing the smoothness parameter and the number of clusters itself. Though having higher adjusted rand index values or ARI values, many of the outputs of SMEB are not visually pleasing when compared to ground truth. In contrast to both of these methods described, our method ScribbleSeg segments by applying a convolutional neural network, and we have the ability to incorporate expert annotation, and we outperform these 2 methods, which are state-of-the-art models. So here is our proposed methodology. This is the algorithm overview of ScribbleSeg. The data is collected from the target tissue slice, in this case human DLPFC sample 151673. There were approximately 3,500 spots and 30,000 gene expression values per spot in this particular sample. So it can be said that each spot has around 30,000 features. And we needed to pre-process these data to reduce this very huge dimension. To do that, we took top 2,000 highly variable genes and then did principal component analysis to choose 15 principal components. And for the case of prior labels of some of the spots, an expert may scribble over the histology image of the tissue slice, or an algorithm like mclust can be used to assign labels at each of the spots. like shown below. Then these scribble annotations, these principal components and the coordinates of each of these spots were passed to our algorithm, and our algorithm produced these outputs. We use an unsupervised segmentation model from computer vision community, which is based on convolutional neural network, or CNN, with 2 hidden layers, filter size of 3 for the first 2 convolutions, and filter size of 1 for the last convolution. After each of the convolution, we do a batch normalization and apply ReLU function as the nonlinearity. Our loss function consists of 3 components. 2 of them are cross entropy losses, and these 2 are related to the similarity and scribble spots. The third loss component is an L1 loss component, which is related to spatial continuity of spots. Here, mu and nu are hyperparameters used for balancing these 3 components. So we show our significant results. Here we show the ScribbleSeg's that ScribbleSeg outperforms state-of-the-art models like Bayes-Space and SCMAB, and also some other spatial and non-spatial algorithms like GMM, Giotto, Leuven, in 9 of the 12 human DLP samples. This comparison is done based on adjusted rand index, in short ARI values. That table, this table shows some quantitative analysis, but when we compare our results with other qualitative with others, like in qualitatively, by visualizing the outputs, we can see ScribbleSeg has the ability to detect the thickness of layer 4 to be more close to the ground truth than Bayes-Space and SCMAB. Also, our output is less noisy than Bayes-Space and SCMAB. For example, we can see some green spots inside the white matter in Bayes-Space's output, but in our case, there are no green spots in the white matter. Here are some results of for other samples. In case of 15670, in the second row, we see that a more visually pleasing output than SCMAB with lower ARI, indicating ARI is not the ideal measure for quantitative analysis. Here we have 0.37 and SCMAB has 0.49, though it is visually not very good. In conclusion, we can say we successfully applied image segmentation methodology of computer vision domain to spatial transcriptomics domain, also incorporating expert knowledge, and we can see a bright future of this work, as a lot of amplifications are possible from this work. We can pre-train our model if more ground truth data are available, and hopefully the semi-supervision present in one of the pipelines will be removed. Thanks a lot.